All right, well, good afternoon, everybody, and welcome to the CPDD Miriam Fischman Memorial Award Lecture. This is an annual award we've been given for, I don't know exactly how many years, but I can tell you every year and every award winner is just fabulous. When I was thinking about this, I was thinking, I have not been on the awards committee, but this must be a terribly difficult decision every year because we have so many great scientists in this organization that fit this bill. And we have another one this year that is this year's award winner. So I'm going to invite Rick Lamb up here to the stage to introduce this year's winner. Rick?
Hello. You might have seen George Bigelow on the schedule to do this, but I'm, as Alan said, not George Bigelow. And George would have been a great person to do this. He's known Kenzie Preston, this year's Miriam Fischman Lecture Award winner, for many years. And he's known and mentored her and worked with her a long time. I feel equally qualified, or at least somewhat qualified, to introduce Kenzie. I've known her perhaps even a little longer than George has. I first encountered Kenzie when we were taking Lou Seiden's psychopharmacology course at the University of Chicago. Kenzie came to Chicago as a graduate student, and she ended up working with Bob Schuster, who you might know. Kenzie was one of a cadre of young, bright graduate students, postdocs, and faculty members that seemed to fill every corner of Chicago at that time. Another one of those was Miriam Fischman. And Kenzie came to Chicago from the University of Illinois at Chicago with a BS in pharmacy. And when she finished her graduate work, she went to Hopkins to work with George Bigelow. And there at Hopkins, Kenzie and George developed procedures, drug discrimination procedures in humans that really formed a translational link between the clinical pharmacology and the preclinical pharmacology of what was then called mixed agonist-antagonist opioids. When Kenzie had completed her postdoc, she was already known as an expert in human behavioral pharmacology and clinical pharmacology and opiate pharmacology. So she just walked across the street and did a fellowship with Don Jasinski and Jack Henningfield. And that's where I really got to know Kenzie a little better, because while we'd interacted as postdocs at Hopkins a little bit, she was over at the Baltimore City Hospital and I was at the medical school, and there's a big part of the city of Baltimore in between. When I went to work with Jack Henningfield, he put us together in an office, so there was only about 3 feet of empty space between us. And I really came to know Kenzie's kindness, her insightfulness, her nurturing nature. In fact, on my first grant application, the two people that helped me most one was Kenzie Preston, the other was Miriam Fischman, who had moved from Chicago to Hopkins in that time. Not shortly, but a while after I'd been at the ARC, Kenzie went back to Hopkins for a brief period. And then when I left, I don't know if there's any connection to this, she came back to the ARC, which then was the NIDA Intramural Research Program. She came back as a section chief. She's now a branch chief. In the years she's been there, she's done some really foundational work on contingency management and some groundbreaking work on using mobile health technologies, both to study drug abuse and potentially to treat it. So it's a real pleasure to introduce this year's Miriam Fischman Lecture Award winner, Kenzie Preston. Now, one thing Kenzie asked me to say before she finishes her lecture, she'll take questions, but it's after she's off the podium, down, and they have to be simple questions that she can answer easily.
We'll get a picture over here.
Okay.
I think. Chris, right here. You want a picture? Yeah. Oh.
Thank you. Don't forget this.
Oh my goodness, Rick, you gave my talk already. So I'm very honored to be here, and especially to give a lecture in honor of Miriam Fischman. And so the title of my talk is In the Footsteps of Fischman. And so as Marion Fischman was a wonderful person, and especially for me, she was a trailblazer and role model. And I chose this shoe as a symbol of that, but I think Marion was a pretty classy person, and I kind of doubt that she would have ever worn that shoe, although some of you who knew her in other contexts could maybe tell me otherwise. So Marion did her graduate work, as Rick mentioned, at the University of Chicago. Her PhD mentors were Bob Schuster and Lou Seiden. And she did some groundbreaking work demonstrating tolerance, development to chronic methamphetamine intoxication, and also showed that methamphetamine produced long-lasting deficits in catecholamine levels in the brain. When I came to the University of Chicago some years later, I had the same graduate advisors, Bob and Lou, and in fact, my dissertation work followed on to what Marion had already done, although I was looking more closely at the acetylcholinergic levels. One of the jobs that I was given was to work with a force lever, which was a manipulandum that rhesus monkey had to reach through a tube and hold it down within a certain amount of force and to earn reinforcers. So I'd have to say that that was probably the least impactful work that I did in my entire career. I don't think anybody ever used the force lever after me, and I don't think that paper was cited maybe more than 1 or 2 times. The good thing about it is that that was the era of the original Star Wars trilogy. And so, in fact, I did have the Force with me in the laboratory. We had Jack Bergman, Richard Fulton, and Suzette Evans. And of course, we had our own Obi-Wan Kenobi in the form of Bob Schuster. So Marion wasn't content to do the groundbreaking work with methamphetamine. She soon followed that with some amazing work with clinical pharmacology of cocaine studies, something that had not been done in decades. You can see here she published them in very high-profile work looking at the pharmacokinetics and pharmacodynamics of cocaine. And in 1982, I got my PhD, and I left Chicago to go to Baltimore, Johns Hopkins, to do a postdoc. And then in 1984, I became a faculty member, and I wrote my first R01 grant, which was funded. And of course, following the lead of Marion, I wrote it on cocaine and did clinical studies, although the focus of mine was on testing possible medications for treatment of cocaine dependence. So in 1984, that same year, Marion also came to Hopkins in Baltimore, so maybe Marion was following in my footsteps, I can't be sure. Richard Fulton, who'd been a fellow graduate student, went to her lab at Hopkins as her first postdoc. And then after some years at Hopkins, both of us left in 1992. Marion went off to Columbia University, and I went to the Addiction Research Center, which soon became the NIDA Intramural Research Program, but not before a little musical chairs. So Ken Silverman, who was Marion's postdoc, came to the ARC and was my first postdoc there. And Richard went to Columbia with Marion, and Suzette Evans, who was a fellow graduate student, was also a postdoc with me at Hopkins. Eventually, I think sometime later, went to Columbia University. So at that point, we no longer tracked each other geographically, but Marion continued to have a very large impact on my career, and I'm sure on many others. She had an impact through her work with cocaine and other stimulants, with studies of opioids and marijuana, and her work has been cited more than 10,000 times. Pretty impressive. She also has had a huge impact on the field through her mentorship. I got this list of names from a paper that Chris Ellen Johansson wrote. You can recognize them as also giants in the addiction field and people who had a big impact here at CPDD. So Marion was truly a trailblazer and role model. And in preparing this talk, it got to me to think about my journey, my research journey, and all the various people that have interacted with and who've helped me and who hopefully I've had an influence on. So my work started after I left University of Chicago. I was doing behavioral pharmacology studies, and I worked with some terrific people there at Hopkins, BPRU. So George Bigelow and I, as Rick also mentioned, developed a human drug discrimination procedure in which we evaluated the opioid agonist-antagonist, which was an earlier approach to producing medications with lower abuse potential. We did precipitated withdrawal studies. So this was a study that we did very early that was looking at the effect of adding naloxone to buprenorphine as a possible way of limiting its abuse liability. We did actual abuse liability studies. This was one with high-dose buprenorphine. We did drug interaction studies. So this was one with Roland and Maxine. Our participants had said that they were boosting their methadone with diazepam, and so this actually looked at that effect of combining those two. And we even did drug self-administration. This was a comparison of self-administration of hydromorphone, lorazepam, and clonidine in people undergoing methadone taper. Then in 1992, Roy Pickens came and asked me to come and work at the Addiction Research Center. And of course, that was intriguing. And he gave me the option. I could do either stick doing the behavioral pharmacology studies, or I could take the scientific lead of a brand new outpatient treatment research program. Well, I had never done a clinical trial, something that was pointed out by George Bigelow in his letter of recommendation for me for that job. But I thought about, maybe what would Marion do? And Bernadine Healy was the director of NIH at that time, I think the first woman. I thought, I'm going to take the challenge, and I will take the scientific lead of the clinical trials section. And so fortunately for me, though, there were really good people there, and we did a series of pharmacological trials. This was one in collaboration with Yvonne Montoya and David Gorelik, testing the effects of buprenorphine maintenance in people who are dually dependent on heroin and cocaine. With Ken Silverman and Annie Umbrecht, we did a series of behavioral trials looking at contingency management and the different parameters associated with delivering that treatment. And with Ed Cohn, we tried out new outcome measures to improve our ability to detect the effects of treatment. Then in 2005, another opportunity raised its head, and that was mobile technology. And this was very exciting and really something we wanted to embrace. And so with my team of collaborators at NIDA IRP, we did a whole series of studies using contingency management where we gave our treatment patients electronic diaries and asked them to report on their mood and activity and exposure to drug triggers as they went about their daily lives. We added GPS measurement, real-time location data collection so that we could look at environmental influences on drug use and mood. And we've studied a series of biosensors for intensive ambulatory monitoring so that we could look at physiological changes that were associated with trigger exposure and mood and craving. So with all of that methodology under our belt, what's the next thing? So we decided that we would apply that methods to translational research. And that was inspired by Udi Gitza, who came to my lab as a postdoc from Yavin Shaham's lab. So Udi was switching from preclinical to clinical. And with Yavin, they were doing studies with the reinstatement model of drug relapse. And in this model, animals are taught to self-administer drugs of abuse. Once that responding for drug became stable, they would take away the drug and there would be extinction such that the responding on the lever decreased down to very low levels. And then once the responding was very low, they would be tested with different stimuli to see what that effect had on their drug-seeking behavior. And what was interesting is that in this model, the same sort of things that trigger relapse in people will also reinstate responding, including drug priming, drug cues, and stress. So this method has been used for screening medications with the idea that medications that block reinstatement of drug seeking might be useful for treatment. And one of the classes of drugs that were particularly intriguing were the alpha-2 adrenergic agonists. These medications like clonidine, lofexidine, guanfacine had been shown to block reinstatement by both stressors and by drug cues. And importantly, it worked across different kinds of drugs of abuse. So both heroin and cocaine and the combination together. The question was, would these drugs work in humans? So we decided to test that, reaching back into our armamentarium of methods and doing a behavioral pharmacology lab study that was designed to evaluate the ability of clonidine to reduce stress-induced and cue-induced heroin and cocaine craving in drug users. So we recruited cocaine users who were not seeking treatment, and we randomly assigned them to one of three doses, 0.1 and 0.2 milligrams of clonidine. And we exposed them afterward to guided imagery scripts of either neutral cues, stress, or drug cues. So this is the result of that study. The outcome measure is cocaine craving rated on a visual analog scale. And this is the group that got the placebo clonidine. And what you can see is that the craving was significantly increased by the stress script and the cue script relative to the neutral script. The group that got the lower dose of clonidine, 0.1 milligrams, we saw a blockade of that response to the stress script, but not the cue script. But at the higher dose of clonidine, we saw a blockade of both response to the stress script and the cue script. So our data from our human lab study looked very similar to the animal studies. And in fact, it had also been replicated with lofexidine for heroin craving by Rajita Sinha. So it does work in the human lab, very exciting. But does it work in the clinic? So we decided to evaluate clonidine for relapse prevention in opioid use disorder. So the question is, if we combine clonidine with buprenorphine, would time to lapse or relapse increase? And we were also interested in how it worked. So would clonidine's effect be exerted by decoupling stress and/or drug cues from lapses? And because we were interested, in the translational aspects of it, we wanted to know whether the foot shocks and the cues that are applied to a rat were analogous to the stressors and cues that heroin users face in the real world. So we designed a randomized clinical trial for clonidine for relapse prevention. We enrolled opioid-dependent treatment seekers, and we tested clonidine 0 milligrams versus 0.3 milligrams per day. So everybody who enrolled in the study began treatment with daily buprenorphine that was delivered in the clinic 7 days a week, as well as weekly individual counseling. Our outcome measure was the result of urine drug screens that were done 3 times a week. And because this was a relapse prevention trial, we had to start off with everybody being abstinent. But these, of course, are outpatients, and they're people seeking treatment for opioid dependence, so they're not going to be abstinent unless we applied another intervention. And so we pulled our contingency management experience out of the bag, and we gave participants vouchers contingent on providing opioid-negative urine specimens to suppress their opioid use. We gave them a quick deadline, and then they had to provide all opioid-negative urine specimens 3 times a week during a 2-week period, and they couldn't miss any urine collection days. And if they met that criteria, they were randomized to either clonidine or placebo. They began clonidine treatment in the clinic along with their buprenorphine. We had a 2-week induction period where we slowly raised the clonidine dose, and then we discontinued the contingent vouchers and continued them on clonidine or placebo for an additional 12 weeks. And then because we were interested in possible mechanisms, we included a measure of EMA, having our participants carry around these electronic diaries with them from the point of randomization to the end of the study. So this is one of the primary outcome measures based on the urine drug screens. This is time to lapse. The days of clonidine and placebo treatment are on the x-axis, and the percent of patients who hadn't lapsed is on the y-axis. And what you can see is the clonidine group in the dark line had significantly longer time to lapse compared to the group that got placebo. This was the other primary outcome measure based on urine drug screens, and it's the longest duration of continuous abstinence. Each of those dots are individual participants, and that line indicates the mean for the 2 groups. And what we found is that indeed the people that got clonidine had significantly longer durations of continuous abstinence, with quite a few of them going through the entire study without any positive, opioid-positive urine specimens. So for our ecological momentary assessment measure, we ask our participants to answer questionnaires at randomly prompted times during their waking hours every day. We asked them to tell us if they'd seen heroin within the last hour, answer yes or no. And we had them rate their heroin craving and their feelings of being stressed on a 4-point scale from no, definitely not, no, maybe, yes, maybe, a yes, definitely I am. So this is the result for the stress and heroin craving. That's a stress rating across the bottom from no, definitely to yes, definitely. And on the y-axis is the percent of participants reporting in the same random prompt that they were craving heroin. So we showed a significant main effect of stress, so that as the higher the stress level, the more likely they were to also be reporting craving heroin. There was a main effect of clonidine. So if we looked at the random prompts overall, the placebo group reported craving in 11.8% of random prompts compared to only 6.3% in the clonidine group. And importantly, there was a drug by stress interaction such that for those intermediate levels of stress ratings, the group that got clonidine were less likely than the placebo group to report craving heroin. Although at the highest level of stress, there was no effect of clonidine. It didn't have any impact on heroin craving at all. And this is the result for drug cues. We saw a main effect of exposure. So when people said that they'd seen heroin in the last hour, they also were more likely to report craving heroin. There was a main effect of clonidine. So overall, the clonidine group was reporting less craving. But there was no evidence for an interaction between drug cue exposure and craving. So if we want to know, does it work? Yes, it looked like clonidine given in conjunction with buprenorphine increased the duration of abstinence and time to lapse. And for the question about the mechanism, whether it worked in humans the same way it worked in animals, I would say that our evidence suggests that this is the case, that the stress and craving were decoupled by clonidine, while cue exposure and craving were not. I think also we can say from this that EMA is a useful tool in addiction research in general, and perhaps in particular in clinical trials if we're interested in more than just overall drug use. So what are the next steps? So we've been working for quite a while to, as many people have, develop the smartphone as a treatment delivery device with the idea that we could collect patient-specific data on the phone, process it either on the phone or perhaps in the cloud somewhere, and then at the appropriate time, deliver a treatment message, a prevention message, or perhaps a health education message. It sounds pretty easy. It is not. There's a lot of things you need to know. You need to know what to collect. You need to know when to deliver that message. And then you also need to know what treatment are you going to deliver. So I'm going to go through each of those. We've actually done the most work on what kind of information should we collect. So in these studies, we've had participants who were enrolled in either buprenorphine or methadone maintenance for their opioid dependence. They've carried these smartphones for long periods of time, up to 32 weeks. And we do compensate them for responding to those questionnaires and for returning the device at the end of the study. We get several different kinds of responses. We get randomly prompted entries, so we're interested in the context. Who are they with? Where are they? What are they doing? We ask them to rate their craving, stress, and mood. And we've gotten a lot of important findings from also asking them about their exposure to drug use triggers. We also have them make entries at important points that we're interested in. So for example, if they've had an intense craving but they didn't use, we've asked them to make entries when they did use, and also when they've had a particularly stressful event. And then we can compare the situation of the events against a baseline that we detect in the random prompts, as well as comparing these things to an objective measure of drug use, which is the 3 times a week urine drug screens. So over time, we've used EMA in 8 different studies. We've collected it from over 1,000 participants, and we've published 28 papers on EMA. And I won't bore you with all of those things today, but I'll hit you with a few highlights. So this is the first study that we conducted. We were interested in seeing if we could document the relapse triggers that Marlatt and Gordon had identified in their relapse taxonomy. And so we looked at the exposure to these triggers in the 5 hours leading up to cocaine use or drug use. And indeed, we were able to show that exposure to drug use increases in the hours before drug use. We were also interested in the overlap of different kinds of drug use. And in this case, we looked at tobacco smoking against cocaine craving. And what we found is the higher the cocaine craving, the more likely people were to also be smoking. And when the event contingents, when they told us they'd craved cocaine and didn't use or actually used cocaine, they had especially high likelihood of also smoking tobacco. So I think this suggests that we need to be treating both of those disorders at the same time. We've done a lot of work with craving. In this study, we showed that cocaine craving increases in the hours prior to cocaine use. We looked at craving during extended periods of active use, so more than 2 weeks of consecutive positive urine specimens, that's that darker line, versus when they were abstinent for at least 2 weeks, and that's the lighter line. And you can see that overall cocaine craving is higher during periods of active use versus abstinence. And most recently, we'd been interested in looking about sensitization to craving, and indeed what we found was that cocaine craving is higher in the presence of both drug cues and stress compared to either one alone. So it was a more than additive effect. We've also been interested in detection of stressful events, and so we developed a new kind of measure with the EMA, and that is we asked people to make an entry anytime they felt stressed, overwhelmed, or anxious more than usual. We asked them how big of a problem was it? Was it just a hassle? Was it a day spoiler, or was it something that would more than spoil their day? And then we had them rate how bad that event was. And this is the results of that. We found that the frequency of stressors varied by severity. So the hassles were more common than the more than day spoilers, which I think is most people's experience. We also found that the numerical ratings matched the verbal description of severity. So hassles were rated as less bad than the things that would be more than spoiling the day. So why were we interested in measuring these? Well, we were interested in seeing if we could detect a temporal relationship between stressful events and drug use. And to do that, we started with our event contingent entries of cocaine use, and we looked back in time. We looked back 3 days in 24-hour intervals. And I would say that the frequency of stress event entries did not vary across those 3 days prior to cocaine use. But what we did find is that the rating of how bad those stressful events were did increase in those 3 days leading up to cocaine use. So, was there a temporal relationship? And the answer is yes, but the fact is it didn't really happen in all of our participants, and it didn't happen for all drug use events. So that led us to publish a paper that was titled "Some of the People, Some of the Time." So that brings us to the question of when should we deliver an intervention? And we've definitely come to the conclusion that as wonderful as EMA data are, they really are not likely to be useful when we're trying to time an intervention. It's a much too burdensome thing on participants. So we needed a measure that would be continuous, and it would be without burden to participants. And so for that, we decided to look to the environment. And there's increasing evidence that our environmental situation affects our health. And that's been shown for all kinds of health conditions, but also for addiction. And so we asked our participants to carry around these little GPS trackers that were developed for photographers to record when they took a picture. But they're about the size of a pager. If any of you remember what a pager looked like. But now we are collecting them on smartphones, much more convenient. So we wanted to know whether there was a relationship between subjective state and environment. And so this is a satellite image of the city of Baltimore. And that blue line is one person's travels during the day through the city. And so we wanted to know, is there an effect on what people experience in the environment on what they report for their mood and activities? And so to do that, we needed a measure of the environment. And so we went to the Neighborhood Inventory for Environmental Typology, and these were data collected in the city of Baltimore by Deborah Furholden, who very graciously gave us access to all of her data. And so we analyzed her data and calculated an environmental score for every 30 by 30 pixel in the city based on the measures that were collected on. And what I have shown here is physical disorder. Blue is low physical disorder, red is high physical disorder. And then for our participants, we calculated an exposure score for every 30 minutes of time as they moved through the city. And we calculated that based on where the pixel fell, the environmental score in the pixel where the GPS points fell, and then we could adjust that based on amount of time. So we could calculate a score for every 30 minutes. And then we did cumulative exposure scores. So we did 0 to 30 minutes before an EMA entry, 0 to 60, 0 to 90, and so forth, out to 12 hours before all of our EMA entries. And what we found is that, indeed, there is a significant association between subjective state and environment, and importantly, the strength of that association increased with the duration of the cumulative track level that we were including in our analysis. And that went out to was maximized at about 5 hours. So what we're trying to do now is to use environmental markers as predictors of drug use, looking at exposure to environmental stressors as well as relapse triggers, with the idea being that we could apply computational models to predict stress and craving, use that information to deliver our just-in-time intervention to prevent drug use. So the way it worked, again, is we're predicting 90 minutes into the future. That's where that green arrow is relative to an EMA. So we need to compare it to something that we know, which is the EMA entry, the craving. We calculate the environmental disorder exposure score, as I mentioned, for every 30 minutes prior to every EMA entry. And then we apply machine learning models. We mostly have done random forest, but we've also done SVM, and we're experimenting with a few others, applying that to 5 hours of time, 6.5 to 1.5 hours before the EMA entry. Our momentary predictors are socioeconomic indicators of GPS tracks, and we're including in that model person-level predictors from the ASI. Because we figure if someone's coming into treatment, that would be like the minimum that we would know about them. And so for our study, our participants carried the trackers and gave EMA data for up to 16 weeks. So we've done a lot of machine learning and doing the 50% training, testing on, say, 50% for the test to see if our algorithm worked. And what we're trying to do now is to simulate, well, how would this work in real time? In this case, predicting likelihood of craving heroin. And so we figured we would actually start with a database of previous patients, and then we would have a new patient who we wanted to enroll in treatment. And to simulate that, we took one person out of our database and then we assigned them as the new patient. So this is a drop one out kind of analysis. And then based on our database, we would calculate the prediction for that new patient for the 16 weeks of treatment, cross-validating it, so comparing it to the actual EMA entries that the person did. Then we would think, well, after a week, we would have a week's worth of data from our new patient. We'd dump that in with our database, and we could calculate for that patient prediction of craving for the next 15 weeks. Same thing with 2 weeks of data, put it in the database, and then calculate prediction for 14 weeks, and so forth, out to using our 15 weeks of data to predict 1 week. And we have done this for each of 187 patients up to 16 weeks. So this is the sensitivity and specificity of our predicted heroin craving. This is the actual versus predicted craving in random prompts. So we, at this point, have 30,000 or almost 40,000 random prompts. We have 1,700 weeks of patient data. Every participants were in there for a mean of 13.6 weeks. And so you can see that for specificity, we got it. It's over 90%. Where we're weak is in the sensitivity, and when we're predicting craving based on no data from an individual patient, we can't predict it all. But adding even a single week of data into that database increases our sensitivity quite a bit. It keeps increasing as we add data from that participant up to, I don't know, around week 8. And so we're somewhere around the 60% sensitivity level. So maybe we're not there yet, but we think we're making a lot of progress toward meeting our goal. So next, in terms of what to deliver, so David Epstein at the IRP is now conducting a trial to test a just-in-time adaptive intervention. His momentary interventions are based on the principles of cognitive behavioral therapy, CBT, and acceptance and commitment therapy, or ACT. And he's using an amazing new study design, which is a micro-randomized trial where he's comparing the response to the CBT messages, the ACT messages versus no message. And this micro-randomized trial is embedded in a conventional randomized between groups trial, so EMA plus just-in-time intervention versus EMA only. So for the micro-randomized trial will give us proximal effects of the intervention, so a few minutes after delivery or a few hours, we can actually see whether that message had any effect. So we can look at the main effect of motivational messaging. And because we're also collecting EMA data in these participants, we will get some more information about the strategy situation fit. And because of the conventional randomized clinical trial, we can look at the distal effects of the intervention, looking weeks to months after the end of that mobile intervention, and we'll be able to look at main effect of interventions on, say, drug use, lapses, and self-efficacy. So I think these mobile phone-based interventions have the promise of being personalized, being cost-effective, and being able to extend the impact of clinician-based treatment. The challenge, of course, is going to be that we have to develop them based on empirical data and test them for efficacy in randomized trials. So I want to end by thanking all the people who've been in my lab over the years. These are graduate students and, not graduates, postdocs, and also our study participants and our treatment staff, the NIDA intramural program for all his support. I want to thank Rick. He already told you my story. We've worked together for a long time. We only ever managed to publish a single paper together, but it's a good one. I would recommend you look it up. And I also want to thank all the many women who've had a positive influence on my career. And I want to call out just a few of them, specifically Chris Ellen Johansson, Harriet DeWitt, Maxine Stitzer, Betty Ty, Annie Umbrecht, Sharon Walsh, and Karen Phillips. And I especially want to thank Marion for forging the path for me and for many of you, and for CPDD for sponsoring this event. Thank you.